Today we'll take up the cell-mediated immune response. In that, first we have to consider the maturation of T cells because cell-mediated immune response is brought about by T cells. That is the T cytotoxic cells, that is CD8 T cells, or T helper cells, that is CD4 T cells. We had done the B cell maturation process. Same way, we have to deal with T cell maturation process. The precursor of the T cell and the B cell both originate in the bone marrow, and their stem cells are same, common. It's only that the cell that has a specialized receptor, that is CD45 receptor, that will remain in the bone marrow to become the B cell. Now, in that lineage of the precursor cell, the cell that possesses CD44 receptor, CD44 receptor, which is supposed to be called as THY receptor, THY means thymus, which is also called as homing receptor, means those cells have to come home. The home of those cells is nothing but thymus. Those precursor T cells which express CD44 receptor are called as CD44 plus T cells or thymocytes. They have not yet become thymocytes, but they have to arrive at thymus. So they will arrive at the thymus. To recall back your memory, we had done thymus structure and function. There is cortex area where all new T cells, that is supposed to be naive, N-A-I-V-E, naive, which are not committed to the antigen yet. They have just arrived from the bone marrow, which are also called as thymocytes. They arrive at the thymus and reside in cortex. In the cortex of the thymus, because of the thymic microenvironment and the nutrition that is provided in the thymus, those CD44 plus T cells or thymocytes starts expressing genes. Until then, they are only with CD44 receptor but naked. They are supposed to be called as naked, or they are not expressed at any characteristic of T cells. Once the thymic microenvironment is given to them, they will start expressing the genes in that area and they become triple positive thymocytes or triple positive precursor T cells. The triple positive precursor T cells, which are also called as immature T cells, express three different types of receptors on them. One set of receptor is called as T cell receptor with CD3 complex. T cell receptor with CD3 complex. Both these receptors, will be talking about it in detail at the end of this cell-mediated immune response. For the time being, you keep in mind the first set of receptor that is characteristic of the T cells is T cell receptor. Just like the way the precursor B cells express their receptor, that is MIGM, that is B cell receptor on their surface. T cells, that is thymocytes, which are now triple positive, which have been exposed to thymic microenvironment, express a gene, and that gene is responsible for T cell receptor complex, which consists of T cell receptor with CD3 complex. The other two receptors are, one is CD4, and that CD4 receptor is a separate receptor plus CD8, that is another receptor. So together with this, that is the T cell receptor complex having CD3, that is one, CD4 is 2, CD8 is 3. So together it is triple positive thymocyte. Triple positive thymocyte. And that triple positive thymocyte is there in the cortex area, now has to come in contact with the thymic epithelial cells. It is mentioned here, cortical epithelial cells in this diagram. Those thymic epithelial cells are the non-professional antigen presenting cells. Okay. In the case of B lymphocyte, we had dealt the process of B cell maturation initial phase is antigen independent phase, whereas in the case of the T cell maturation, it is antigen dependent phase. They have to be presented with antigen, and the thymic epithelial cells express both class 1 and class 2 antigens, and by expressing both class 1 and class 2 antigens, and these antigens that are expressed on their surface, this is class 1 and this is class 2. So the antigens are self antigens. The class 1 as well as class 2 both express self antigens. The reason for expressing self antigens is for the positive selection of the T lymphocytes. The T cells, the precursor T cells, first have to learn the self antigens and not to develop immune response against the self antigens. Therefore, they are first exposed to the self antigens on class 1 and class 2 MHC. That is by the thymic epithelial cells. Let us see what happens in the positive selection. Now, this process of contact of the precursor T cells with the thymic epithelial cells, which are expressing the self antigen, is called as positive selection. If the class 1 MHC expresses the self antigen and the precursor triple positive thymocyte comes in contact with the class 1 MHC through its T cell receptor, the T cell receptor establishes the synapse or contact with the class 1 MHC, and the antigen presented in the class 1 MHC antigen presenting groove is the self antigen. And the receptor, that is CD8 receptor, comes in contact with the alpha 3 domain of the class 1 MHC. It is a must. So, class 1 MHC is expressing self antigen. TCR establishes the contact with the class 1 MHC, that is synapse. As soon as that is established, that precursor thymocyte, which has expressed CD8 receptor like this, the CD8 receptor which is there on its surface, will establish contact with the alpha 3 domain of the class 1. So once that synapse has taken place, that T cell, that is precursor T cell, is committed for class 1 MHC. Okay, so this triple positive thymocyte, which established first contact with the class 1 MHC, together with its CD8 receptor contacting alpha 3 domain, now will become double positive thymocyte. In the sense, it will lose the CD4 receptor. The CD4 receptor does not have affinity to class 1 MHC. It will lose. It will retain only TCR complex with CD8. Okay, so that is the cell that will now in future will become the CD8 T cell. CD8 T cell, the one which establishes the first contact with class 1 MHC, will commit itself to become the CD8 T cell. Excuse me. Now the second one, same precursor T cell, another one which is there in the clone, will establish contact with the thymic epithelial cell through class 2 MHC. The class 2 MHC is presenting the self antigen. The TCR complex establishes the contact with class 2 MHC. Simultaneously, the CD4 receptor establishes the contact with the beta 2 domain of the class 2 MHC. That precursor cell, which has come in contact with the class 2 MHC, will establish simultaneously contact with the beta 2 domain of the class 2 MHC through CD4. Now, this triple positive thymocyte will become double positive by losing CD8 receptor. And this will retain the CD4 receptor, and that cell which has now established contact with class 2 MHC commits itself to become the CD4 T cell. The CD4 T cells are going to become T helper cells. And those cells that come in contact with the class 1 MHC will become T cytotoxic cells. This process of selection by self antigen on class 1 MHC is called as positive selection, and without this T cells are not going to mature. And this process of presenting the antigen on the class 2 MHC to the T cells is called as MHC restriction. Means if the class 1 MHC is presenting the antigen, that will restrict the T cell to become only TC cell or CD8 T cell. That is T cytotoxic cell. If the antigen is presented on class 2 MHC, that will restrict the T cell to become TH cell or CD4 T cell. TH is T helper cell. So please keep this in mind. Without the presentation of antigens on class 1 or class 2 MHC, T cell maturation is not possible. And the positive selection of T lymphocytes or T cells takes place in the thymus, and that is done through thymic epithelial cells, and they present the self antigens on class 1 and class 2 MHC, and class 1 restricted precursor T cells or thymocytes will become CD8 T cells, and class 2 MHC restricted T cells will become TH cells. Now let us presume that the bands that are there in the cortex area, if they do not come in contact with the thymic epithelial cells like this, within a week's time, if they don't come in contact with the thymic epithelial cells, those T cells are going to go for apoptosis. They will be committing suicide like this. Apoptosis means they are not coming in contact with the antigen. Of course, they have expressed both TCR CD3 complex plus CD4 as well as CD8, and they have become triple positive thymocytes, but they could not establish contact with the self antigen presented by class 1 MHC or class 2 MHC within a week's time. So they are supposed to be killed by apoptosis. They undergo apoptosis. But those which came in contact with the thymic epithelial cells, they will survive and they will go through the positive selection. Now, once they have come in contact with the self antigen on the class 1 MHC or class 2 MHC, they are committed to become CD4 or CD8 T cells. Now, they are expected to now leave the thymus and migrate to the lymph node. Leave the thymus and migrate to the lymph node. And go there and look for non-self antigen. In the lymph node, they will be presented with non-self antigen. Or in the medulla of the thymus, there are macrophages and there are dendritic cells. Okay, interdigitating dendritic cells. If they have the non-self antigens on them, means they had processed the antigen and by chance they were there in the medulla region, they will be presenting non-self antigens to the TC cell or TH cell that has been committed by the positive selection. But very rare instance that in the medulla of the thymus, the macrophage and dendritic cells will be presenting non-self antigens. They will be presenting self antigens only. Now let us see what happens if they present the self antigens. So those T cells which were selected to become CD8 and the CD4 T cells will come in contact with the self antigen presented by the macrophage or dendritic cells. So if it is the self antigen once again presented by class 1 MHC by the macrophage or the dendritic cell, that CD8 T cell which had emerged by positive selection will now undergo apoptosis. It cannot come in contact with the self antigen once again. First, it had come in contact with the self antigen presented by the epithelial cell. It got selected as CD8 T cell. Now, it has to come in contact with the non-self antigen, but if it comes in contact with the self antigen and this process repeats, if it happens, then those cells are supposed to be undergoing apoptosis. They cannot, for the second time, come in contact with the self antigen. And if they repeatedly come in contact with the self antigens, large number of cells that were selected as CD8 T cells or CD4 T cells will be going through apoptotic process. The CD4 T cells have to come in contact with the non-self antigen on the class 2 MHC. If they don't come in contact with the non-self antigen, they will also undergo apoptosis. So this process may happen in the medulla region of the thymus, where those T cells which had committed themselves to become CD8 and CD4 T cells descending down here, and they are supposed to escape from the thymus and go to the lymph node to explore the foreign antigen or non-self antigen. But by chance, if they come across the antigen presenting cells presenting the self antigen and they come in contact with them through their receptor, they will undergo apoptosis. Otherwise, in the lymph node, when they reside there in the cortex, when they arrive and they migrate to the paracortex area, from there, they migrate to the paracortex area. In the paracortex area, they have to come in contact with the antigen presenting cell, the professional antigen presenting cell, which has to present the non-self antigen, if at all non-self antigen is there. If the non-self antigen is not presented, those T lymphocytes within a week's time will undergo apoptosis. Those which were selected as CD4 T cells and CD8 T cells, if they don't come in contact with the non-self antigen in the lymph node, they will undergo apoptosis. But they do not remain in the lymph node for a long time. Every 48 hours, those thymocytes that is selected CD4 or CD8 T cells, which go to the periphery, that is to the lymph node, they have to come home. They become homesick. They have to come home to the thymus within 48 hours. Once again, get charged means get some nutrition from the thymus and go back to the lymph node. And that cycle will be repeated. Coming to thymus, going back to lymph node, waiting for the non-self antigen. If they don't get the antigen, they have to come back. But those which come in contact with the self antigen, they will die there in the lymph node itself. But those which do not come in contact with the antigen, they come back to the thymus. That cycle of repetition, that is coming back to thymus, going back to lymph nodes, coming back to thymus, going back to lymph node, repeats for at least about a week. And within a week's time, if they have not come in contact with the non-self antigen, those selected thymocytes also should die. They will undergo apoptosis. But let us see what happens if the selected T lymphocyte, that is, let us take for the example, first example, that is CD4 cell, that is the cell that was selected as Th cell, which had come in contact with the class 2 antigen. Now is selected and it has come into the lymph node and establishes the contact with the antigen presenting cell. It establishes the contact with the antigen presenting cell. So the antigen presenting cell is presenting a non-self antigen in the class 2 MHC antigen presenting group, which was processed by that antigen presenting cell. So that T lymphocyte, Th cell, CD4, has come in contact with that antigen in the class 2 MHC through its TCR, T cell receptor, that complex. As soon as it establishes that synapse, the CD4, which had established contact with the beta 2 domain of the class 2 MHC, once again establishes contact with that. So this synapse of the CD4 T cell through its TCR complex with the class 2 MHC, simultaneously CD4 complex beta 2 domain of the class 2 MHC, results in signal transduction 1. And that signal transduction 1 results in positive signal for the T lymphocyte to undergo activation. And the first gene to be switched on will be the IL-2 cytokine gene together with IL-2 receptor gene. So IL-2 cytokine gene, IL-2 is supposed to be T cell growth factor or T cell mitotic factor. It is an autocrine factor, means it is produced by the Th cell, that is T helper cell or CD4 cell, and it acts on that CD4 cell itself, produced by the same cell, acts on itself, is called as autocrine. Here in this diagram, it is shown. So with the result, that Th cell now being activated by the presentation of non-self antigen on the class 2 MHC by switching on the gene for IL-2 receptor undergoes mitosis. In that process, that synapse is sufficient. It will leave that antigen presenting cell. It will express the second another receptor, that is CD28. And the CD28 is the second receptor, important receptor expressed by the activated Th cell, will establish contact with the B7 receptor of the antigen presenting cell. Okay, and that will result in signal 2. And when that synapse is completed, that T cell has to dislodge itself from the antigen presenting cell. These two signals will be more than sufficient to make that Th cell competent for that non-self antigen. With the result that Th cell now activated because of that non-self antigen leaves that antigen presenting cell, and now it starts undergoing proliferation. That proliferation of the T cell, CD4 T cell, the mitotic activity results in production of clones of T cells. Clones means same T cell committed to the same antigen. That Th cell, which is committed to that antigen on class 2 MHC now, undergoes repeated proliferation, produces millions of clones, and that CD4 T cell selected for that antigen will also now program its gene in such a way that it will put the TCR, a best fit TCR, which will fit into that antigenic receptor very well. Okay, and it will now commit itself to become effector T cell. Effector is shown here as E, and memory T cell. The effector T cells will remain in the lymph node, which are clones of T cells committed to one particular antigen. And these effector T cells now have to delineate or have to now differentiate into two different types. Whereas the memory T cell that was formed for committing to that antigen will migrate to the bone marrow. The memory T cell will stop secreting the IL-2. It will become IL-2 minus. It will not proliferate anymore. It will migrate to the bone marrow and reside there. And it will be there as a memory T cell, that is CD4 T cell, for that particular antigen which it had come in contact for the first time, presented by the antigen presenting cell. Okay, and the effector T cell, which is formed, will now express another receptor, that is CD40L receptor, which will come in contact with the CD40 receptor by the antigen presenting cell, and by then it has put in the TCR, T cell receptor, to the best fit for that antigen. Second time it will come in contact with the antigen presenting cell to become the final TH cell to differentiate itself into TH1 and TH2. Okay, initially formed CD4 T cell is called as TH0. 0 means it is not that it is not doing any functions. It is already activated against the antigen like this. This you can call it as TH0. It has not yet expressed CD40 receptor. It was first committing itself to the non-self antigen. And with the expression of two receptors, that is expression of two signal transductions, one through the TCR complex, another through CD28 receptor. This committed TH cell now has to differentiate to become effector cell, and that effector cell will become TH1 and TH2. When it is becoming TH1 and TH2, it has to come in contact with the antigen presenting cell through the second receptor, that is with the CD40L. This is TH cell which expresses CD40. Again, comes in contact with the antigen presenting cell which is expressing CD40 receptor. Once that is done, this TH cell has now the process of differentiation, and that process of differentiation results in two lineages of the T cells, TH1 and TH2. The TH1 CD4 cell or TH1 cell produces the cytokines which will be IL-2, interferon gamma, and TGF beta. Please note, it will produce only these three types of cytokines. TH1 cell, which has descended as an effector cell from the THO or TH0 cell, will now start producing the cytokine IL-2, interferon gamma, and TGF beta. And this TH cell, TH1, is responsible for the maintenance of the memory cell, formation of the TH cell dependent hypersensitivity, that is called as DTH response, which is type 4 allergic response, and that Th1 cell is producing interferon gamma as well as TGF beta. Both are inflammatory cytokines. So Th1 cell is responsible for type 4 hypersensitivity, which we'll be talking in unit 4. Plus, it is also responsible for inflammatory cytokines and inflammatory response, immune response. Whereas Th2 cell will start producing the cytokines such as IL-4, IL-5, IL-6 and IL-10. Please note IL-4, IL-5, IL-6 and IL-10. Interleukin 4, 5, 6 and 10. Interleukin 10 is also considered to be an immunosuppressive cytokine, which is not expressed in the beginning. Once interleukin 10 is expressed, the immune response has to go down. Suppression of immune response takes place. And when does it happen? It happens only when the antigen load in the body has gone down or antigen load has become negative. There is no more antigen in the body. If there is no more antigen in the body, the immune response has to be suppressed. There is no point in producing more immune cells, or there is no point in producing more antibody. For that reason, both cell-mediated immune response as well as humoral immune response should be suppressed. Therefore, it is a regulatory cytokine, so IL-10 will suppress both these responses. Okay, whereas IL-4, IL-5 and IL-6, we had come across these cytokines when we spoke about B cell differentiation and B cell formation of memory cells. And therefore, Th2 cell, which has differentiated cell from the Th0 cell, is responsible for activation of B lymphocytes as well as differentiation of B lymphocytes. The Th2 cell is responsible for controlling the humoral immune response. And the humoral immune response is nothing but production of the antibody. Antibody of 5 different classes are produced because of the influence of the Th cell. And that too, it is Th2 cell. Now you know the T helper cell is very important for not only the cell-mediated immune response as well as humoral immune response. If the Th cell does not exist, or if you block the Th cell maturation process, or if you block Th cell activation process, you can completely suppress the immune response. That is both cell-mediated immune response as well as the humoral immune response, both can be suppressed. And that is what happens in an immune deficiency disease, that is severe combined immunodeficiency, SCID, severe combined immunodeficiency. In the severe combined immunodeficiency, the patient suffers from deficiency of CD4 T cell. That is, T helper cells do not exist in them. For some genetic reason, T helper cells are not going to be produced. If the T helper cells are not going to be produced, that person will suffer from both severe combined, means both cell-mediated immune response as well as humoral immune response will not be there. In such a condition, survival of the person will be very, very difficult. They are more prone to all sorts of bacterial and viral infections. Therefore, these people can be saved only through passive immunity by giving them the antibodies produced in other people, or by bone marrow transplant, which will produce healthy Th cells. Since they have a genetic deficiency where they cannot produce normal Th cells, therefore they are suffering from severe combined immunodeficiency. They have to be transplanted with healthy bone marrow, which is capable of producing the Th cell, in the sense the lineage of Th cells, and they should have intact thymus. And if they don't have thymus, then also they will suffer from combined immunodeficiency. So the two possibilities are there. If the thymus is not developed, the Th cells will not be produced, or T lymphocytes will not be undergoing maturation. Then also they will suffer from severe combined immunodeficiency. Other possibility is they may not have normal healthy Th cells. In that case, they will also suffer from severe combined immunodeficiency, because humoral immune response is always Th cell dependent, and I've already talked about that. T cell dependent B cell activation, which will give immune memory for the B cells. If the T cell independent activation of B cells takes place, there is no immune memory, and that immunity is not long lasting. So you know the importance of now Th cells, and Th cells which lineate themselves into Th1 and Th2 play different roles, of which Th2 cells are specially meant for humoral immune response. Means they are responsible for activation and differentiation of the B lymphocyte, because they secrete the cytokines responsible for activation and differentiation of B lymphocytes. Whereas Th1 cells are meant for inflammation. The reason being, they are the major contributors of inflammatory cytokines, that is interferon gamma and TGF beta. Both are responsible for severe inflammation. Probably the severe inflammation experienced by the virally infected people, the two coronavirus infected people, and these two are the cytokines which are causing the damage in their body. Okay, then what happens next? So those T lymphocytes which had become committed to the CD4 as well as CD8 T cells. Now those CD8 T cells, okay, how to look for now cells that are expressing non-self antigens on their surface. And those cells which will express non-self antigens, if they are virally infected, or if they have intracellular bacteria, intracellular pathogen, and those cells will be either normal other somatic cells like epithelial cells or whatever, or they could be even the macrophage, because macrophage, when it is phagocytizing a bacteria through the endocytic process or simple phagocytic process, which I had mentioned that there is a possibility that the bacteria which goes inside the macrophage after phagocytosis will prevent the macrophage from killing that bacteria by prevention of the endosome from fusion with the lysosome. And I had mentioned one of the bacteria that has the capability is tuberculosis bacteria. The tuberculosis bacteria, when it is engulfed by the macrophage, will remain inside the macrophage without getting destructed or destroyed by preventing the fusion of lysosome with the endosome. And it will grow in the macrophage by proliferating inside the endosomal vesicle by utilizing all the nutrition and nutrients provided by the macrophage. And such a macrophage becomes the granuloma, and it remains in the same place where it had engulfed that bacteria, it will be unable to move from that area. And such a macrophage can present by accident the antigens of the tuberculosis bacteria on class 1 MHC to the TC cell, or the macrophage can engulf a virus, and there are many viruses which will prevent the macrophage from lysing them or killing them. In same way, they may present the antigens of the viral origin on class 1 MHC. We'll be talking about this presentation of the antigens of the intracellular pathogens on class 1 MHC separately in antigen processing and presentation after this cell mediated immune response. Okay, then there is a possibility that epithelial cells of different tissue origin might be infected by the virus or bacteria. Those antigens also may be presented by class 1 MHC, and such cells will be recognized by the TC cells, and they will be responsible for killing those cells, and that mechanism we'll see in the next class. But the response of the TC cell will depend on the TH cell cytokine, that is TH1 cell, which secretes the IL-2 that is responsible for the activation of TC cell. So we'll see that in the next class. Thank you.